Okay, thank you, Dr. Hina. Good day, Bismillah Rahman Rahim. Assalamu alaikum and good day, ladies and gentlemen. Thank you very much for joining us today for this webinar, which will be delivered by Professor Dr. Mustafa Guzel. We are very delighted and honored to have you with us today, sir. The topic of his webinar is Story of Favicovir, Domestic Drug Development for COVID-19 Treatment. And before introducing him in detail, I would request Professor Dr. Iqbal Chaudhry, Coordinator General Comstech and Director ICCBS, to please welcome our guest speaker for today. Over to you, sir.
Bismillah ar-Rahman ar-Rahim. Good morning and good afternoon. Assalamu alaikum, ladies and gentlemen. Distinguished participants, I can see some old friends, very prominent scientists in the field of chemistry and medicinal chemistry are here. And this actually represents the popularity of Dr. Mustafa Guzel. Also, the quality of science he's conducting. He's one of the leading chemists of OIC region, and certainly one of the most prominent scientists in Turkey, who has contributed immensely in the field of drug discovery and development. Dr. Mustafa is not only a dear friend, but also a very good human being who touches your heart in multitude of ways. His science, his personality, his creativity, and his connection always help many people, individuals, institutions, and the country. And that was actually happened during COVID-19. He was at the forefront of Turkish science, handling a pandemic of an unprecedented magnitude. Mustafa and his colleagues in Turkey has done remarkable contributions, made remarkable contribution in forwarding science to handle this major health crisis. Dr. Mustafa Guzel has worked at the frontiers of drug discovery and development. He made a seminal contribution not only in developing of new chemical method, but also linking laboratory chemistry with industry. I think this is a real contour of his research, that he has been able to link with the industry in a major way. He has optimized the synthesis of many very important drugs. He indigenized technologies involved for the synthesis of drugs in Turkey, and he has added the innovation of efficient synthesis of lots of very important life-saving drugs, including antiviral, in Turkey and elsewhere. He has trained lots of people from developing world. His laboratory is kind of a center, focal point where lots of people from different countries come and learn. We are so absolutely delighted to welcome Dr. Mustafa Guzel in this comstech Distinguished Scholars series. We select the best of the best in the OIC region and beyond. And we are so fortunate to have that Dr. Mustafa has accepted our invitation. So thank you very much, Dr. Mustafa Guzel. And thank you very much, everyone, for being part of this exciting learning experience.
Thank you very much, sir, for your kind words. And in addition to what has already been said about our speaker today, let me introduce to you all again. Dr. Guzel graduated from Hacettepe University in 1987. He completed his PhD from Clemson University. And in 2014, he started working as an assistant professor at Istanbul Medipol University. In 2019, he was appointed as the Department Chair of Molecular Medicine and Biotechnology. In January 2020, he then became the Director of Drug Discovery Development Research Center in a newly established Health Sciences and Technologies Research Institute, and is currently leading 2 critical drug development projects for COVID-19 with a local pharma company, as well as several domestic and European granted drug discovery projects in the field of molecular discovery and development laboratories of the universities. And with this, over to you, Professor Guzel, for your lecture.
Thank you very much. Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Peace and blessings be upon you and all of the attendees. First of all, I would like to thank Professor Iqbal and the team and all ICCPS team inviting me for this nice lecture. Hopefully it will be nice. And today is, as you know, Laylatul Regaib, and I am also congratulating you to this blessing the day and the night. And so we started the peaceful tree, including the Ramadan. I wish you a happy Laylatul Regaib also. So I would like to actually address this lecture. I'm trying to go to the next slide if I can. No, okay. So what I would like to do, I would like to address this lecture as actually interactive as possible. So if there are some questions, and please feel free to address at any time, especially the young students. And also, I would like to make this not too scientific, if I may. So, of course, there are some scientific aspects of it. So I will give you the feedback of what we did, what we accomplished within a short period of time during these COVID times. The title of the project that I want to focus on, development of the drugs and the raw materials and the new synthetic methods and the formulations to reduce external dependence against pandemic virus, which is COVID-19. As you know, this is the project that I was leading at Medipol University, and Atabay is the pharma partner. So I would like to address this. This is the seminar outline. I don't want to too much squeeze on, but I will give you some current situation analysis, give you the feedback of what I look at the introduction of the drug discovery and development, and industrial production of the Favipiravir, and some other projects, acknowledgements, and conclusion remarks. Okay, so I have this thing with me. I don't know why. Not that I'm gonna gain a Nobel Prize in the future, but I have this thing, pictures with the Nobel Prizes. So this is the Turkish and only Turkish scientist who won the Nobel Prize in 2015. So he's our private scientist, one of the prominent scientists in the United States, and one of the Turkish American scientists, Aziz Sancar. He's in the Chapel Hill professor, biochemistry professor in the School of Medicine. So I was lucky to meet him in about 2-3 hours, and also present a kind of placard from our university, and discuss the details about what we can do in our country in terms of his findings in circadian clock and photolysed chemistry. And so these are the pictures that I want to share with you before I go into our lecture. And this is E.J. Corey, Harvard professor in 1990, when I was, of course, young. 1980, I was doing a PhD, later stages. 2001, Nobel Prize shortlist, K-Berry shortlist. And this is 2010 to Negishi, Purdue professor, again, Nobel Prize in chemistry, Negishi. Actually, I was lucky to hold his Nobel Prize medallion with my hand, because we had a dinner together. And 2012 Nobel Prize winner, Brian Kobilka, Stanford professor, and also Martin Chalfie, 2008 Nobel Prize winner in chemistry. Again, he is a Columbia University professor, and he was actually the father of GFP, green fluorescent protein. And with that, a lot of right now scientific articles come from the GFP, as you know. I don't know why these two pictures are in the place, but this is in 1982, first time I entered actually the organic chemistry lab we were trying to extract fragrance from the actually oranges with that picture. So this picture is we are trying to do diabetic research with our drug in Remar here. So I don't know why these pictures are here, but I wanted to share it with the youngsters. So there are some survey in 2019, global trust in professions. Actually, who do we trust in and what professions are these? Okay, and here is the link of the PDF. When I look at the proficiencies, mostly scientists we trust, and public mostly trust in scientists, then the doctors, then the teachers, and after that, armed forces, the police, then the list goes on. Okay, and here are the dispersion or distribution in the countries. So again, scientists in the top, then the doctors, then the teachers go on, and later on, armed forces like police, and all these goes on the list. By country, so in Turkey is the fourth country after Russia, Argentina, and Mexico. 70% of the people, public, trust scientists in the world. Okay, and in 12 countries among them, Argentina, Germany, Hungary, South Korea, Saudi Arabia, Mexico, Poland, Russia, including Sweden and Turkey, they trust scientists, what they say, okay, 12 countries, and the doctors, that second profession. Interestingly enough, teachers are also in Brazil and USA, they are the top profession that they trust. And of course, we don't have to say, needless to say, on all countries where they are asked, politicians are considered the most untrusted professions, because sometimes, of course, what they promise is not actually coming into picture, and they don't trust on this. Not that I want to go into politics, but this is what the outcome is. So when it comes to the drug discovery, and as you know, it requires significant infrastructure and resources. But I say that it's not impossible. If we dream, of course, it can become a reality. So when I look at this, I am in this part of this picture, basically the blue part. Pre-discovery, you have to synthesize around 10,000 compounds. If you're lucky, you get in vivo toxicology animal studies. And if you're lucky, then you get investigation new drug, IND submit. And then you start those escalating studies, which we call safety studies, which phase 1, with the 2-3 cohort is good enough, like 20-30 patients. Then phase 2 is efficacy and safety studies. The NAS phase 2 is split in phase 2A and B. And then if you are lucky, then most of the time it goes to in the phase 3. Now phase 3 also split into phase 3A and B. What is the difference? Basically, you do this in different countries, different ethnics, different groups. Like you do it in Mozambique, you do it in Canada, Finland, so on and so forth. And again, if you are lucky, you go in the new drug application submission, and then you go in this one approved drug. So it's not easy. So I mean, you can probably see my hair's all gray and white. I have been in this business almost 25 years. I synthesized with my own hands around 12,000 compounds. These are all, by the way, novel compounds. Did I get lucky to become all of them actually to this one dot? 12 of them drug candidates. Remember, drug candidates, not drug yet. So I have phase 2-3 Alzheimer drug, one is in phase 3, 2-3 diabetes drug in phase 3 that I was actually contributed to part of it. So I mean, it's not easy to bring this compound in the market. That's what I'm trying to say. But still, once you bring the compound in the market, your job is not done. Still, you have the post-marketing surveillance, process development, scale-ups, and so on and so forth. So the federal government and industry has a huge role in R&D. This is the 2004 numbers or the 6 numbers. So the NIH around $30 billion pays, mostly the basic research. In the translation research, usually the governments and the federal governments do not support, and the clinical research almost nothing. So the private sector or the private entities usually support the clinical research. In terms of drug development, this is how the picture. So these two triangles complement each other. This is what I am trying to say. And I like this slide also. Why? Because these are the 12 big pharma company that how much they paid for the R&D within the 15 years, and how many drugs entities came into picture, basically the launched, or we call this marketed drugs. So if you look at this, AstraZeneca in the first, not that I blame them, by the way, they spent almost, these are, by the way, $1 billion. They spend almost $60 billion. They bring 5 compounds in the market. Whereas Amgen spent almost half of them, $30 billion, and they enter or launch 9 medicines. So R&D, it's a huge investment. You have to pay a lot of money, and look at the numbers. Only 9 compounds you can. But for example, Novartis becomes a wiser decision in terms of they spend less money, but they bring more compounds on the launch. So so for us, countries like these, Turkey and the Pakistan and developing countries, so we have only one bullet basically in terms of medicine development. So we have to be really careful to actually spend our resources and discuss this carefully. So these are the statistical, basically the bars. So the 1% of the actually the medicines that we try to develop actually becomes the reality on the registration. In the discovery stage, 19% of them, so only becomes 1% of them becomes a reality. Another interesting fact that I want to mention is 90% of the drugs that enter in the clinical trials to make it to market. And as probably most of you know, average cost of to bring the market right now, estimated cost around 2.5, 2.6 billion dollars. It's not easy, and it takes 15 years. So as a medicinal chemist, I teach pharmacology, introduction to clinics, organic chemistry and medicinal chemistry, but I consider myself as a medicinal chemist. So as a medicinal chemist, we should realize that innovation process is really difficult because the disease target is complex. The body, sometimes the absorption is poor. Sometimes the toxicologic profile is really high because there is the small window between the therapeutic window we call TD50 therapeutic dose and the ED50 or the LD50 little dose. So we should realize the most compounds that we make do not become medicines. I consider those compounds basically, you know, the maze. Sometimes if you know where to go or where not to go, it's better to go. It's better to realize to actually you go much faster to the exit actually. So as I give you the example at the beginning, I synthesized with my hands throughout my career 12,000 compounds. Only 12 of them just become drug candidate, not drug yet. Only this medicine that I wanna, this Favicovir, except this is the generic drug, which we will highlight a minute later. Okay, so another important aspects, these are the scientific disciplines and the development capabilities that basically the experts to make the drug discovery and development reality. The biochemist has to be there, experts, molecular biologists, cell biologists, physiologists, in vitro, in vivo pharmacologists, structural biologists, bioinformatics, computational chemists, computational biologists, and biophysics, medicinal chemists like me, genomics, so the list goes on. So if you have made a compound and you have a clinical outcome, 5,000 patients, and if you don't have a biostatistics, clinical statisticians, then you will look at the data and data will look at you. So all these experts has to stick together. Sometimes that's what I see the problem in Turkey. I don't know how it is in Pakistan. These experts should stick together. These scientists, they have to devote their time basically to develop molecule. So our project was, as you know, when the world hit by this pandemic coronavirus, we started also get back and investigate the literature. What should we do? How can we help our country? These three medicines were the top, of course, COVID-19 treatment. Of course, there are right now new drugs, as you know, the Favipiravir. There are some right now steroid also. Therapy is also like dexamethasone and cortisol also for the intubated patients. But if you have a fever and you are a COVID positive, usually these three medicines are prescribed. So the Favipiravir is the first one. The hydroxychloroquine, right now there are some ups and downs. The clinical data is just now coming for this because there are some cardiovascular risks. They are saying that there might be. Remdesivir is another one. As you know, this is the patented and protected. These two are not. So we started working on this. So I'm going to focus on mostly the Favipiravir synthesis because for the sake of time, Favipiravir, as you know, it's discovered by Atomo Chemicals. Trade name is Avigan, 200 milligram tablet, and it's prescribed for COVID-19 in Turkey. First day dose is, we call it actually heavy dose loading, 1600 milligram. Then the second day, 3 doses that they use, 3 by morning, 3. So you finish it in 5 days. What is it good for? Basically, it works for the RNA viruses, different influenza, including rhinoviruses, bunyaviruses, West Nile viruses, and different viruses. This was basically influenza medicine, as you know. How does it work? Its mechanism of action is RNA-dependent polymerase inhibitor. So you cannot use it for a pregnant woman because it's teratogenic and embryotoxic. So it's an also reminder. We should be very careful. How does it work? Favipiravir basically tautomerizes with the enzyme in our body. It phosphorylates. It becomes the active form in the ribose phosphate. Then it basically stops the proliferation of the virus in the plasma. And this is the hydroxyxanthine guanine phosphorotransferase. This is the protein that's basically maybe the only picture in the PDB data bank that I remember. So that is the mechanism. How do we synthesize the Favipiravir? This is the stepwise synthesis of the Favipiravir, starting from the pyrazine carboxylic acid, 3 amino pyrazine carboxylic acid. Basically, this is the steps. I don't want to go in detail, but you make the first esterification. After that, MBS, you brominate. After bromination, you convert the amine to OH. Now, after that, the ester becomes an amide. With the POCl3, you basically chlorinate the bromine and OH. With the KF2BAP, you convert that to nitrile to difluoro compound. And after that, you move on to one-part synthesis of Favipiravir. Although you see the different attitude, different approaches, what we did, we designed a one path synthesis without isolating the intermediates. So our goal was, since the COVID-19, the patients was, as you know, exploding, increasing numbers, our attitude or the approach was, we have to investigate very fast with the process development and optimize these methods with the industrial scale. That was our goal. So this is a true university and industry collaboration with the government incentive. So how can you hear me? Am I sound good? No problem, right?
Yes, very loud and clear.
Okay, all right, thank you. And these are the pictures and the melting points and basically physicochemical properties of all the intermediates that we highlighted. Why I'm showing this? Still in, I don't know, this is a generic molecule only. This is not a zero, the new molecule, novel molecule, I must admit. But some people still don't believe that we can make this in our hand, in our house. So I am showing that as a proof that at least in a lab scale and also in the industrial scale, we can make this. It's a for us, it's a piece of cake. If you're a medicinal chemist, mostly, of course, you have to have the spectroscopic data. These are the intermediate data that I want to emphasize without boring you too much. Intermediate 3, intermediate 4. These are the NMR data. Intermediate 5, moving on, and the HPLC data. And also we compare it and also with the authentic the chemistry papers by Louis on the NMR data. We compared and made sure that that our data is in line with the literature data, carbon-13, proton NMR. Then after that, we started focusing on how can we improve, because we needed more bulk of this, right, Favipiravir. We tried to do the DMF pathway. DMF pathway was not very useful for us. We investigated different methods. One path synthesis with the pathway 1. We made the sodium salt and different extraction methods, and increased the yield to around 30 to around 50, 55 now. Made different salts of it. Instead of toluene extraction, we did ethyl acetate extraction. So all of these, as you know, it's not an easy job. If you are doing it at a large scale, and also you need to do this in the reactors also later on. So we did this in a short period of time. These are the results. I don't want to bore you too much. Method optimization, of course. Still, there are some steps that we want to optimize the rods using different acids, different conditions. These are the HPLC results and NMR results of the compared all the spectroscopic data. Overall, we were able to, with the one path synthesis of pathway 1, around 43. So within 40 days, I would say, we were successful enough to bring this to industrial scale. Then we got our approval and the licensing by the Minister of Health, because we needed to make sure that we have enough quantity domestically. So by the results, we did 8 steps route the target product of Favipiravir and a lab, as well as the industrial scale, fully synthesized with complete domestic force and facilities. The structure of the target product was determined by known analysis. Optimization study have been completed for each step. Monthly production capacity aimed right now around 3 to 4 tons. Industrial scale production and alternative synthesis, of course, methodologies is still ongoing. Science is forever, I would say. So we still keep investigating each steps and each to, of course, for industrial scale, time is essence, and also as well as the even if you can save 2-3% for each step, that's a huge amount. So this is what we're trying to do. With my assistants, we did basically a photo album, and we gave it to the President Tayyip Erdogan. So these are the pictures that I want to share with you without boring you too much. So this was the project with the TÜBİTAK and Ministry of Science and Development and Ministry of Industry. And of course, as I said, this was the project with Medipol and Atabay together. In June 12th, President Erdogan said that our scientist was able to synthesize domestically this medicine under the umbrella of TÜBİTAK with, again, as I said, Medipol and Atabay consortium. So in July 10, we were able to get our license with our project. This is the picture that that we threw. These are the pictures that in the photo album. In 22 April, sorry, these are Turkish because these are PDFs. I apologize, but I am trying to translate it to you in English. Because I could not change the, because I gave these talks and also in several occasions. So I must apologize for you that these are English. These are not English because it's a PDF files. So I could not translate them. I apologize. So, and in the 3rd of May, we entered the lab because we started receiving chemicals. And 6th May, TÜBİTAK president, we had the Zoom meeting. These are the pictures that in the lab. So even including me, I was making here in the Father's Day, I was away from, I don't want to do agitation, but basically we stayed in the hotel with the 12 people working day and night, helping, try to help our country. These are the pictures and the proofs that actually we are working day and night. So the 12 people, within 40 days, we were able to successfully produce this first Favicovir, then the hydroxychloroquine, of course, remdesivir. At least we were trying to make that, although it's not licensable, as you know, it's patent protected, but we wanted to make it that we can at least be comfortable. And in March 2020, I am telling Minister of Health that we are gonna work on this COVID-19 project. And 13th of June, we visited Minister of Industry, providing these boxes of our medicines, informing him that we were successful. Then Minister of Trading, also we even discussed this exporting opportunities of this medicine. Right now we got the exporting license for this because we gave enough, around 6 million boxes, we gave enough quantity to Ministry of Health. At the beginning, 10,000 boxes were donated to Ministry of Health for immediate use. And 17 July, Minister of TÜBİTAK, sorry, the TÜBİTAK President, Professor Mandal, visited us to see us how we are doing and in terms of scale up. These are the pictures. I apologize again. These are Turkish media, social media, how we can actually we did contribution in science, especially for the treatment of COVID-19. So I'm gonna skip through these. In August 9, President Erdogan visited us all the COVID platform, TÜBİTAK COVID platform. So I gave this photo album to him. We were lucky enough to meet him. At least I thanked him to supporting this and being a part of this, basically the consortium. Within 2-3 weeks, he also invited us and meet us, discussed basically in detail what we can do and improve our basically drug discovery and development infrastructure and how we can help our country. So we talked about around 1 hour what we can do. And this is the team that I always thank at the end of my talks. These are the 12 people that basically we were able to make this medicine become a reality. Of course, we have several projects for COVID-19. This is another one of them that I want to just give you the highlights of it, not details. We have a montelukast is one of these leukotriene antagonists, as you know. It's a asthma drug approach, and we are investigating treatment for this for COVID-19. We entered this in the clinical trials right now. We patented for this indication because it was not patented for this indication. And right now we submitted also in the bioarchive the articles because we saw some pseudo-neutralization and SPR assays that we can really show the antiviral effects of this medicine. So we have another project, maybe this will interest Professor Iqbal and Hina. This is an isolation and synthesis of novel medicine, molecule analogs, especially in artemisinin annua and some other medicine plants that we are interested in. Right now, we show some good antiviral activity and for COVID-19. We are preparing the manuscript right now for this second project. And we have some additional collaboration projects that this is another one. Remember, I showed you the cryptochrome with Aziz Sanjar. So with his pioneers, especially the postdocs, he's a professor at Koç University. We discovered a molecule that destabilized the cryptochrome. We submitted this manuscript. We showed some in vivo efficacy with this. And more in publications, and this is another publication with Istanbul University, with thiazole moiety, with antifungal activity. This is another one, silver nanoparticles with some antiviral microbial activity. Another one, this is the leishmaniasis research that we published recently. More on, we did some cancer research collaboration with Professor Hasan Korkaya in United States. This is the patent. And we published 2 out of this research with Nature Communications and an oncogene in anti-cancer research. Another one, again, this is a Parkinson disease drug development. This is the patent that we, international patent that we submitted. This is the leishmaniasis patent. I don't want to bore you too much. More research on, again, Parkinson drug development. Another research. And this is the GBM, glioblastoma multiforme, Cape, probably propolis. This is one of the molecule that belongs or in the contents of the propolis, caffeic acid phenethylester. So we made some analogs of it. More on this methyljasmonate, jasmine family. We investigated the anti-cancer activity of this in our research with the cost relation. So these are the collaboration projects. So what we do in terms of research, which I want to highlight, these are the research that we do: cancer and cancer metabolism, metabolic diseases and diabetes and obesity, neurodegenerative diseases like Alzheimer, Parkinson, and Huntington disease, autoimmune diseases and inflammation. So we can do drug discovery, hit identification, lead optimization, and development, of course, SAR, PKPD, and ADME properties. And these are the 9 projects that we did here. Of course, we are open to all energetic and motivated researchers, PhD and doctoral researchers who are willing to join our group, and always welcome. Please send their CV to me, my mail, mguzal@medipol.edu.tr. So I'm going to conclude within 2-3 minutes, then I will go to the questions and answers, if I may. So this is the beautiful Istanbul, Bosphorus. This is where we actually located. which I will show you in the next slides. This is Medipol right now near the Bosphorus, basically. This is where we are. Around 30,000 people right now get education here. This is the International School of Medicine, that site, and the Center for the Drug Discovery and Development. And what we can do in the Medipol University, in the Health and Sciences and Research Institute, we can do basic and translational research like organ and disease and dish, regenerative and restorative medicine research, some neuroscience. We can do a lot of course, investigation and biological active molecules and herbal preparations, host microbiome interactions, and all sort of these things that we can do and collaborate with. So in the Health Sciences and Technology Research Institute, we have 5 centers, which cancer research centers. Again, I apologize, this is a Turkish, but I'm gonna just quickly translate to you in English. We have Medicine Research Center, or the Drug Research Center, which I am responsible for, Cancer Research Center, Regenerative and Restorative Research Center, then Biomedical and Biotechnology Research Center, and then the Neuroscience Research Center. And most of these yellow labs, the core labs, are basically in picture. We have it, and we are trying to open a couple of more labs in future, hopefully. I always, at the end of my lecture, of course, thank to my team. We have 3 assistant professors in our group in the Drug Discovery and the Development Center, and with the Güzel Research Group. We have several postdoc researchers, at least 7, 8 of them. I always thank them. We have 6 PhD students and 5 master students. We have, of course, almost 4 more group members. We always thank them, and a lot of undergrads. So we always thank to them. Without them, it's a team effort. We cannot, of course, become a successful. This was the Favicovir team that we were able to become the within a short period of time. So I truly thank to my team. Of course, I thank to the president of university, Professor Sabahattin Aydın. Now he became a vice minister of health recently, and also thank to the president of Sabita. He is the guy that actually brought me from the United States. The collaborators, always we thank to them, especially Adil Mardinoğlu. He's the ex NIH president. He is still professor in King's College and bioinformatics in the KTH. William Bill Gmeiner, and all these and several collaborators, we thank them. Of course, funding resources, we thank always Medipol University, Scientific Committee, and TÜBİTAK, Sci Life, COST, European Corporation, Science and Technology. And so with that too much in mind, I want to at least thank to inviting me, the ICCPH and the HEG team, inviting me, and hopefully we will see you guys in Karachi, and I want to invite you in Istanbul. We are still dynamically in talk with Professor Iqbal and Hina. How can we basically bring our sources and forces together and to basically do the scientific research together? A last word of advice for the youngsters that I want to say: science is in a 3 span. Those who learn about the first span become arrogant. Those who learn the second span will be humble. Those who learn about the third span realize that they don't actually know anything. This is Kadni Ibn Tayyib's word. The second word from Charles Darwin. I a little bit tweaked these words. Science and technology. He said science and art, but I switched it to science and technology, are like 2 wings of the bird. Societies that can use these two wings can easily fly and become free. Those who cannot fly become chickens. The chicken society does not even realize that their eggs are taken from behind while picking a handful of a bait thrown in front of it. I mean, I guess you probably get this advice, and hopefully we don't become a chicken society. Without further ado, I want to thank you for your attention. And Turkey and Pakistan, always two sister countries. So we want to bring and share this scientific attitude and scientific collaboration together with Medipol University, with our infrastructure, with the University of Karachi, to basically help infrastructure and to develop our togetherness, especially in the bad times. Because I remember that there was an earthquake in Turkey. The first country that called was Pakistan. We had the same thing. Pakistan had some earthquake or some other natural disasters. I remember that the first country that always connect is Turkey. So let's keep this relationship live and fruitful. And again, without further too much talking, I want to thank you again for inviting me, giving this opportunity, and discussing, and at least showing our within a short period of time our results. And thank you again. If there are some questions on the students or audience, please feel free to ask.
Thank you very much, Professor Ghazel, and thank you to you for accepting our invitation and for this very wonderful talk. We have 3 questions in our chat box. Let me read them to you. Number 1 is from Dr. Shahid Ahmed.
Maybe I can see you also. I'm going to stop share if you guys need it, so I can see you guys. Okay. Please go ahead, Kazima.
Right. So number 1 question is from Dr. Shahid Ahmed. He's asking, does this drug Favicovir is suitable for COVID-19 mutant, which is being reported in UK and other countries?
Good question. Actually, we have also some mutations in our country. I don't know about the numbers right now. They are saying it's around 100. It's been used, yes, since this is antiviral. It is showing some efficacy, but do we have the real clinical evidence? Not yet.
Right.
So actually, the numbers has to be more. So we have to see really. In order for me to say like really truly that drug works for these mutations, I need to see at least a handful of clinical outcomes. Like let's say 1,000 patients, we have treated this amount of mutated effects or the mutated patients treatment and show a good efficacy. So I don't think we are there yet, even in the world.
Right. So another question from Dr. Ishtiaq. He's asking, what is the efficacy of Favicovir and does it affect based on seasonality?
What is efficacy in terms of in the COVID-19?
Well, I think yes. Do we have Dr. Ishtiaq with us if he would like to elaborate on his question?
If I may answer the efficacy, if he's asking, I don't think he's asking the animal efficacy. This drug is it's also being used in the Europe as well as I think heavily in Turkey and the Middle East MENA region. So it's showing really good efficacy in terms of if you are, for example, COVID-19 positive, you have a you're beginning at the beginning, you have the fever. What I believe, if you don't take this medicine, because I've seen it with my eyes, several patients did not take this medicine. They thought that, oh, it's too much you're taking because 8 tablets in the morning, 8 tablets in the after at night. not with the empty stomach, with the, of course, with the full stomach. So, and I've seen patients getting really big benefit. I think what I believe, it blocks the viral load and entering the virus to the lungs. If you start taking the medicine early stages, but if you are intubated and you are like viruses in the lung and it's proliferated there, for this drug to become efficacious, is really not, chances are slim. Still, it works. I heard some other cases, because I have talked to a clinician in Medipol University. He treated around 40,000-50,000 COVID-19. When they look at the BT scan, they can clearly tell, okay, this patient needs not hormone treatment or the cortisol treatment or the dexamethasone. This patient needs this and this and that. So this antiviral, if you detect the COVID positive PCR test early, and you are like at the beginning stages, because as you know, there are, getting exposure with the 50,000 cells and 50 million cells of the virus makes a huge difference, right? So if you are a COVID positive and early stages, you caught it and you take Favicovir, I know that it works, because this is the first line of treatment in Turkey. I don't know if I answered the question right or is sufficient.
Okay, thank you, Dr.
Thank you, Professor.
I think Nafis, or I forgot his name.
Yeah, Dr. Ishtiaq, is it you? You can please talk.
Please, please.
Yeah, okay, thank you, Professor. That's very clear. And just about my question is, the next one is about the seasonality. So is there any difference from Turkey or some other countries about this seasonality effect? Thank you.
You mean bioavailability? I couldn't hear your, what do you mean by seasonality? I didn't understand.
I think he's talking about different seasons. Like, would it be different?
Oh, seasonality. Okay. Well, for COVID-19, I don't think there are a lot of misconceptions that we heard. We were thinking that in the summertime, it's going to go away. It's going to fly. It's going to become like a, let's say, common immunity, right? For all the cultures on Earth, but it didn't. So there are still unpredicted pathways of this virus. We still don't know, a lot of things unknowns. So with different seasons, with different terms, in the winter time, of course, you're becoming more in the congested areas, especially in the weather becomes colder, especially in the common or the areas. People get exposed with a lot of, in the outside, in the hut, you don't get exposed much. So in the seasonal, therapeutic effect might be different, but virus is still there. I don't know if I
thank you.
As well.
Thank
yes.
you, Professor Guzel. Well, I would welcome our audience to question directly from Professor Guzel. We have 2 more minutes to go.
Excuse me, let me clear, because since we have restricted the mic, so I think it's better to write your question in the chat box.
All right. So let me read out some more questions that we have.
Sure.
So Dr. Farnaz Mumtaz is asking, kindly tell us about the resistance profile of this drug.
Tell us what? I'm sorry?
Resistance profile of this drug. If this drug is found to be resistant to some.
I mean, some clinicians still don't believe this medicine that it works. Okay, but in Turkey, we had like almost 2.5 million right now PCR positive tests, and I am sure at least more than 1 million, maybe 2 million people are treated with this medicine, and some of them they said it's too much. This medicine, it doesn't work. Some people don't believe in it, but I believe that because clinical evidence also shows that there is this medicine works. Does it have the resistance? Yes, because especially this neem mutated, it may have some resistance, like happens in the also cancer drugs. You might have some resistance because these virus cells, they are not dumb. I mean, they find a way to survive, right? With a different pathway. So once you load with this medicine, because don't take me wrong, I mean, first day you take actually 3.2 gram of this medicine, 16 milligram, 1600 milligram in the morning, 1600 milligram afternoon or at the evening. So it's too much. But and I don't think this medicine is toxicology profile is also, I look at this different species, its bioavailability and the toxicology profile is very good. So no side effects so far, even the 3.2 grams. So is there any truly resistance? That also numbers needs to come. Still, again, with that numbers, I cannot say there's a really drug resistance with this medicine, but we need to see more data.
Thank you, Professor Gozel. I think we can take one last quick question. We have a question from Mr. Muhammad Adil Abbas. He's asking, have you observed any side effects of this drug?
This, as I said at the beginning, this is a teratogenic and embryotoxic. So that's probably the most important thing. So the pregnant woman cannot take it, and also her partner cannot take it. So the side effects, and not that adverse side effects that I know. That, I mean, as far as I know, there is no risk of cardiotoxicity. There's no other, at least proven toxicity effect that I know.
Thank you, Professor Gozel. With this, we come to end of this webinar. Thank you very much, everyone, for joining us. For everyone's information, this lecture is recorded and would be available at our YouTube channel of Comstech, and link will be on our webpage for this event. And with this, thank you again, Professor Gozel, for this wonderful talk and sparing your time.